Good afternoon and welcome to the CRISP Winter Virtual Meeting. I'm John Thompson, the Catskill Regional Invasive Species Partnership Director, and today is December 10th. You should be seeing the agenda on your screen, and before we get too far along, I'll ask each of you to introduce yourself in the chat so that we have an idea who's here today. And just to mention our meeting etiquette here, please mute yourselves as feedback and background noise can interfere with others who are speaking. Closed captioning is available at the bottom of your screen. You can hover over the dots at the bottom of the screen and click more, and then you can click on the CC symbol to get closed captioning. As you can see from the agenda, I'll give some updates for CRISP. We'll get to hear from organizations that we have subcontracts with in 2024, Otsego County Conservation Association and Cornell Cooperative Extension, Columbia and Greene counties. Meredith Taylor from New York City DEP will give an update on the discovery of fishhook water flea and the reservoirs this year. I'll present our forest regeneration survey that we had sent out. We'll finish with an opportunity for any partners or organizations that are in the meeting to give updates, and I expect that we'll finish up about 3 o'clock. So for CRISP updates, we've started working on the 2024 CRISP annual report. Some of the impressive metrics include that during 2024, the CRISP aquatic and terrestrial strike teams surveyed a total of 3,408 acres of water bodies and uplands and managed a total of 219 acres in the CRISP region. In 2024, CRISP led a total of 35 events for 791 participants. 21 trainings were offered for identification, reporting, early detection, control, and prevention of invasive species. Other programs raised awareness about spotted lanternfly, beach leaf disease, and other emerging species. We've been working on fact sheets, and we've created over 90 fact sheets so far for 90 different species. So those will be providing them as we get requests for information. We'll be posting them, links to them on the website. So we'll have a lot more information becoming available as time goes on in the next month or so. I would imagine that we'd be putting those up on the website. We released the CRISP cooperation agreement in 2023. Organizations signing the agreement are agreeing to support the mission of CRISP in performing invasive species management. They get points on the request for proposals, and they may be considered to be nominated for the CRISP steering committee. We had the following organizations sign on in 2024, including the Adirondack Research, Flying Trillium Gardens and Preserve, Greene County Soil and Water Conservation District, Lake Association of White Lake, and Sullivan County Soil and Water Conservation District. And thank you for all of those organizations who have signed on and other ones that had signed on previously from our steering committee. If you're interested in the agreement, we have it posted on our website, and I'd be happy to chat with anyone about it. Alexa Tamburello unfortunately had to leave her volunteer and outreach coordinator position this year for medical reasons. The job is now open for applications, and we're in the interview process. Hopefully we'll get somebody on board early in the new year. We have a number of programs scheduled for 2025, and we'll be leading volunteer work days at the Ashokan Rail Trail and Callicoon Riverside Park. Dan Snyder NERP has a webinar that's scheduled for February 24th. That webinar will be on beach leaf disease and spotted lanternfly, and it will occur during National Invasive Species Awareness Week. We have exciting news that I'd like to share right now, and that's the announcement of the 2025 CRISP request for proposals. We have $18,900 available in our subcontracts, and details for the RFP are on our website. That's www.catskillinvasives.com, and I think Sarah will put that link for us in the chat so everyone could see it. And let's see, and I'll stop sharing. And if Jeff O'Hanley is online, we can get started with the presentations that we have on the agenda.
I am.
Oh, great.
Just got here.
Great.
So let me just pull this here.
So, as I mentioned, we'll have a few of our subcontracts. Jeff and the Otsego County Conservation Association were awarded the 2024 RFP for the Otsego County Aquatic Invasive Rapid Response Team. So that's what Jeff will be talking about. And take it away, Jeff.
All right. Well, thanks so much, John, and apologize for kind of dropping in here after things got started. It's called overbooking myself.
Yeah, that's no problem.
So, and thanks everybody for putting up with me today. Again, as John said, Jeff O'Handley, I'm the program director for OCCA, and this is our project for 2024. I'll just give you some quick background for those of you who don't know who OCCA is. We are a private nonprofit environmental organization founded in 1968. We cover a broad range of environmental topics in Otsego County and slightly beyond the borders of Otsego County as well. And just going back, our original history when we were just in, strictly volunteer organization or a staff of one, most of our invasive species work was in sort of public information and getting the word out as a 40-plus-year-old newsletter about water chestnuts in there. As we picked up staff and had more capacity, we started getting involved with volunteer projects with invasive species removal. In 2015, we received a 3-year grant from DEC, our first invasive species grant from DEC, and that helped us fund watershed stewards at Otsego and Canandaigua lakes, and also helped provide funds for boat wash stations at both of those locations. And then in 2018, we received a second DEC grant that allowed us to have to do early detection work for aquatic invasives. And this was, I think, a little before CRISP actually had aquatics people on board. So we had people for 3 years going out doing surveys out in assorted water bodies in Otsego County, which was great. But the one question that came up all the time was, what do you do when you find one? And in some cases, our people could get out there and do, maybe a little bit of removal or, contact property owners in the area if it was a privately owned water body, IMAP it, et cetera. But, usually what we would end up doing is trying to organize some volunteers and get some folks out there. So the impetus for the rapid response group came about. I'll give a lot of credit to Paul Lord, who pushed us a lot to kind of get involved in this. And the idea was, again, to basically have a response team, in our area. And we kind of got started initially with this team last year in 2023. And you may remember I did a short presentation on it last year as well. And the purpose of this team was that it would be flexible, would be able to respond to new infestations, and also to be able to support some of our ongoing control efforts in other locations with previously established populations. And we thought that it was pretty successful, but we did learn some lessons from it last year, and those were that I needed to start sooner because we kind of got a late start on it last year. We didn't get our first folks out in the water in 2023 probably until mid-July. We also recognized that we needed to have some consistent and regular days instead of what was sort of an on-the-fly scheduling, and that I needed to overhire because we were working with people, and if we needed, say, 4 people to do a job in a particular place, we needed to have 7 available because inevitably people would drop out or not be able to work, in part because of those inconsistencies in the schedule. Uh, So coming into this year, we lessons applied. Um, After we put in our RFP, we also kind of looked things over a little bit, and we decided that one of the things we needed to do was to change the job parameters a little bit and to change the job description. Where we had been going with sort of per diem people last year, we wanted part-time, so more a more permanent employee. We also decided to set up a consistent, regular schedule. Our team was able to, we were able to get funding to provide our team for 2 days a week, and we set up a schedule of Tuesdays and Thursdays from early June through late August. Um, and I figured that I didn't necessarily want to make people work 2 days in a row in some of these settings. We also had a little bit of extra cash available so that our team could support some of the weekend work we would do with volunteers, some of our regular, water chestnut pull in Goodyear Lake type of thing. So they were able to pick up a couple of extra hours here or there by supporting those efforts as well. And so we were fortunate that and I started earlier. Heading into this spring, we had our two rapid response team members, Colin Gilbert on the left, who is a SUNY Oneonta student, Ray Hovis on the right, who is from Vassar College, both interested in natural sciences and environment and getting out and helping to make a difference. So they came in, we did some training with them, and they hit the water on the first Tuesday of June. Okay. Over the course, and I should point out they were also supplemented by our summer intern, Erin Rock, who, in addition to doing some invasive species work, she also did education and filled in other program areas. And then either myself or our watershed facilitator, Will, was out with the team pretty much every day that they were out there. So from beginning of June to the end of August, when they had to go back to school, we had a total of about 29, not about, we had 29 work days out there, 10 sites in the county and just out of the county where the stars are. These were pretty much, and these were full days, full work days. In total, I calculated about 82 and a half acres of water surface that they did work on. Um, Estimating in excess of 10,000 pounds of invasive plant material was removed, and the three species that we worked on the most were European frogbit and yellow floating heart, which I believe those are both tier 2 species in crisp, and water chestnut, that old standard that we just can't seem to get rid of. Okay. And I want to kind of zero in on two different areas that they worked in. One was the yellow floating heart. I get shivers just seeing this because this is a real beast. There is right outside of Otsego County in the Crisp region is about a half-acre pond in South Worcester that say greater than 75% coverage. I think that when this was first reported, it was probably 85% or more of the surface area that's covered with yellow floating heart. Sarah Coney had come to us in September of 2022 to talk about us getting involved with this. And in 2023, we were out there for, I think, oh, yeah, look at that, 5 visits with the team in 2023, and we maybe worked in about a third of this pond. In 2024, we were back and we did 11 total visits, and we got through about half the pond. And, if you kind of look at that and say, that's it. This is a beast. We probably could have had the team in here every single day of the summer, and we may have not gotten through it anyway. Just to show you what it looks like, this was the pond on August 1st in 2023, before our first visit, or at the start of our first visit. Um, Aside from the emergence that you see here and there, almost everything here is yellow floating heart. There's a couple of patches of water lilies out there. Um, but other than that, it's just about all yellow floating heart. And just to show you, this was we treated last year about, as I said, probably up to, if you can see where my cursor is going, probably up to around here. When we came back at the beginning of June, it was heartening and disheartening at the same time. Heartening because we actually did see some open water here, and we thought maybe we were effective. Disheartening because a lot of it had also filled in with that floating heart. So June 6th, this was the start. We got in here. And then on August 29th, um, this is what we left behind. I will say that pretty much every time the team was in here, they had to go back and clean up stuff close to where we started. We just have to kind of work our way out as plants grew, filled in a little bit. And if you've never worked with this stuff, like I said, it's a bit of a beast. It's a real tough plant to deal with. Just in terms of area covered again in 2023, this was the approximate area that we had treated. As I said, about a third of this pond. Um, and this is estimate. This may be slightly overestimated. I'm not sure. At the end of 2024, I actually walked out with GPS and took points pretty much over the sort of the edge of our treatment area. Um, it starts out fast. When you're in this narrow area of the pond, you clean through it really fast, but the further out you go, the tougher it gets in some ways. Okay. And we did a weight estimate. We This stuff was heavy. I think this made up the bulk of the 10,000 plus pounds of plant material that we pulled. Okay. But that was the yellow floating heart. I also wanted to mention the water chestnut in a small pond in Oneonta as well. This is a half-acre privately owned pond just outside the city of Oneonta, and this is a good example of how this team can work in a rapid response situation. I think it was on October 2nd, a notification came in from IMAP Invasives that there was a new infestation of water chestnut. And it turns out that the property owner himself reported it. I think this is a picture from the IMAP entry, in fact. I don't know how many years it was there before he finally decided to report it. But anyway, my estimate was about 2,500 square feet that was pretty much just fully covered with water chestnut. And then there were plants scattered throughout the remainder of this pond. Sarah and Will, I got to go on vacation right when we started this. This is, again, early August. Sarah and Will went out and talked to the property owner and looked at the site. And then the team got out, I think it was about 2.5 days of work. Again, I missed the first 2 days. By the time I got there, we were down to probably about a 250 square foot area. But again, it was super thick stuff. But I'm glad to say that we were able to give the team a victory. So this is Colin and Ray, and that's Aaron, our intern. Um, one of the things about dealing with invasives, of course, is that you can feel like you've never actually beaten them. They're still there and everything. And I know that next year if we go back here, this pond will probably have plenty of water chestnut again. But it was nice to actually be able to pull the last water chestnut from a pond and give them kind of a victory late in the season. So just in terms of looking ahead, I think we found that this model worked pretty well, having the consistency of a team, consistent people, consistent days of the week, consistent hours. The only thing that would change is I might say, okay, next on Tuesday we're going to go to, do yellow floating heart, or we might change it up depending on new things like going to this pond in Oneonta. I will say that participation, coordination, and cooperation with the aquatics team at CRISP is vital. We exchanged a lot of information. They came out in the field a number of times to support us and would also help point us in the right direction, places we should go. So that is a key part of success of any response team in the region. For my sake, I will say that better documentation is still needed. There's a lot of lack of photographs this year. We did our pretty well with IMAP reports, but some of the could always use a little bit better documentation on this, I think. And was there anything else? Oh, yeah, it actually turned out to be a little bit more expensive than we thought. Don't worry, John, we didn't bill you for anything. But we found particularly it was mileage for us this year that the cost of going back and forth to some of the different sites was a little bit more expensive than we thought. So future years we're looking for funding again for the future, but we would build that in. So And I think, my slides are not advancing, but that was it. So, oh, there we go. Again, we appreciate the funding through CRISP, as well as support that we received from the Watershed Stewards Program and SUNY Oneonta, as well as the Scriven Foundation. And if anybody has any questions, I'm happy to answer them.
Great, thanks, Jeff. I think, did we already pay your invoice? So
Yes, you
maybe
did.
didn't cover your mileage. Sorry about that.
Oh no,
Um,
that's what are you gonna do? Mileage was expensive this year.
yes, it is. Yeah. Um, so I guess a question I was thinking about. So if you're able to go ahead with it next year, are you going back to the same sites? Are there new sites that you might go to, or it
Well,
happens to us
certainly
where we have sites
the
that we give up because
certainly the site at for the Yellow Floating Heart is one we would get back to. Um, it's personal. It's personal there. And some of it depends. This year there were a couple of sites where we went to and expected we would have to go two or three times, and found that um, like we did a Larchwood Lake in Lawrence that has a water chestnut problem. We went back there, and I figured we'd have to be there three or four times this summer, and I think we went for 2 half days because it was not as bad as we expected. Um, so you kind of sub in new places as needed, and take out other places. We looked at Summit Lake up in Springfield, and the frog bit up there is a lot worse than we realized, and I think that would be a place where we would probably clear where the boats go in and out, as opposed to
Mhm.
trying to hit the whole thing, because it just doesn't, necessarily make sense. That's what kind of we did up at Weaver Lake as well, um, out of the crisp region, but in the watershed. So we
Right.
went up there a couple of times and cleared out around the launch site. And again, the good thing about this model was that it was pretty flexible. So if we found that it only took a half day at one site, we'd say, okay, let's go over, we'd go over to Goodyear Lake and keep working on that, or we'd go back to one of the other sites and work there instead.
Great.
Yep. And
Uh,
as Sarah pointed out, we dumped that floating heart on dry ground.
right. Are there other questions? Do people have other questions or comments for Jeff?
I'll just say how incredibly useful the project is, if for treating aquatic invasive species in CRISP. OCCA and I'm sure other organizations might have had lower volunteer numbers than would have been appreciated. We've seen that this summer, unfortunately, so we would not have gotten nearly as much done if we were just relying on volunteer power or just the strike team. So fantastic.
Yeah. Yeah, thank you. Yeah, that was good because we were able to supplement. We had a few days where we had decent numbers of volunteers, but there were other days where it's like, oh, here's the one person plus, a couple of the strike team people. So, you know, yeah, there was definitely some issues with volunteers this year.
And I guess I could mention just generally for the 2025 RFP, work with tier one and tier two species is always priority.
Yep.
Um, and in addition, I can mention that we've incorporated some of the group concept mapping statements from our strategic planning a few years ago. So you'll notice some of that, some of those topics in the RFP as being higher priority.
Yep.
So
Less water chestnut, more frog bit.
yeah, well, hopefully we could have less frog bit over time too, right?
Yeah, that would be nice.
Are there any other questions? Otherwise, I guess,
John, I have a quick question.
oh yeah, go Mark.
I was just Googling frogbit to learn a little bit more about it and see that it's for sale everywhere. Um, is this the aquarium plant that is for sale everywhere online? And I'm just wondering if in the the paper you're going to give, or the description of how to manage it, there would be like warnings in it for people, how to let the public know a little bit more about what to do if you dump your aquarium somewhere, that kind of thing.
Sarah.
Yeah. So the frogvid that's typically sold for aquariums is a different species, different genus even. It's Linobium labigatum, which sounds like a spell. That one's actually native to North America and South America, I believe. Or actually, no, I think it's Linobium spongium that we have up here in North America. So those are actually native. This one is introduced from Europe. I believe it was introduced as an ornamental at first. So it isn't currently sold in the aquarium trade, but it's not unreasonable that somebody would go to a pond and get it. We are working with the Upper Susquehanna Coalition. They got some funding that we can put up signage at these specific launches, because several of these launches aren't DEC launches. They're public launches, but they're not state launches. That we can put up frogbit specific signage at these launches. We're warning people that it's here, don't move it around.
Great. Yeah, the signage element is great. And the public education element.
Becky, do you want to start sharing your slides, and maybe are there any other questions for Jeff? Or actually didn't give people an opportunity to ask me any questions about the CRISP in 2024, but if anybody has a question right now, I'd be happy to entertain it. Or a question for Jeff.
Okay. I will say thank you.
Thank you, Jeff. And yeah, next, I guess we could start with or move on to Becky Palmatier. She's at Cornell Cooperative Extension, Columbia Greene counties. And CCE's been working on CRISP invasive species outreach for a number of years. I think almost every year since 2013 or 2014. And hopefully you can share your slides. Okay.
Can everybody see them?
That looks like that looks good. Thank you,
Okay.
Becky.
All right. I was just making sure I did it.
Yeah.
Correct. Zoom updates, and then now I'm like, okay, what am I doing? So
Right. It's always changing where controls are. Yeah.
Okay. Hi everyone. I'm Becky Palmatier with Cornell Cooperative Extension of Columbia Greene counties. I'm the program director. Like John said, we've actually been subcontracting for a number of years on doing the outreach and education for CRISP. So some of what I talk about, it's probably the stuff I've talked about before, but we see a lot of success with kind of the model that we're doing. So kind of how it works is that we act as the main subcontract, and then we subcontract the subcontract to CCE Delaware, Schoharie-Otsego, Sullivan, and Ulster counties. So we're really getting a good kind of overview of every area.
More crap in there.
So Columbia Greene maintains the website and the hotline. Honestly, the hotline is mainly manned and answered by our master gardeners who have been trained. And then we host all the CCEs in the partnership, host educational workshops, again, taught by a lot of master gardeners, and kind of new this year, we got our master forest owners. So CCE Columbia Greene kind of is the coordinator for the regional master forest owner program as well. So we got them trained so that when they can go out and do forest visits on people's property, they're helping identify the invasive species and kind of talk about management a little bit. And then every one of the partner CCEs exhibits a static display at their fairs and at other educational outreach events, but the main one for Cornell Cooperative Extension in general is our fairs. So this is kind of the website, and I just kind of put the link on how to connect to it. All right, so I already mentioned we do a number of educational workshops. This is some pictures from our partner agencies, associations, and they vary. So sometimes the topic is a specific invasive species. Sometimes the topic is an overarching thing on invasive species. Sometimes the education is actually a part of a larger program. So I already mentioned like the Master Forest owners might be doing this as part of their entire woods walk. This year, actually, we had volunteers from Regeneron come to our own model forest in Acre, and we gave a presentation on invasive species, and then actually had them go into the forest with us and help pull some. I believe it was mostly stilt grass, and there was an aquatic too, but I can't remember which one it was at this time that we had them help with. So every CCE kind of handles it differently, but then they report it all back to us, and then I do a final report that's shared and given to John at CRISP. And then, like I said, the other main thing we do is some type of static display at different educational outlets or the fairs. So most CCEs, if you've never gone to a county fair, we usually have a very large presence, probably a whole building that's all ours. That gives us a lot of leeway to put up very nice big displays on topics like invasive species. And then at least CC Columbia and Green, we usually have staff on hand that people will come and like ask questions, or our master gardeners are always there to answer questions. So it's really just a great opportunity to reach a large number of people. All right, so that's actually the end of my slideshow. I move fairly quick. Like I said, we've been doing this quite a long time with CRISP, and this is kind of what we do every year. So I can take any questions anybody has. Or did I leave anything out, John, for this year?
Well, I got distracted a bit. Maybe you mentioned it. But do you know how many people like see your displays at fairs that you show up at?
So like, for example, our numbers for Columbia and Greene counties, Columbia County, we estimate the foot traffic to be about 6,000 people that move through our building. And then at the Greene County Youth Fair, there's about 3,000. So that's kind of probably Columbia is more in line with the rest of the CCEs. Greene counties tends to be pretty small as far as fairs are concerned. And I did forget to mention that actually we in Acre, our Greene County office, we actually maintain a static display that's always up. So we always have that display available for people who come in, which is really good because we have the model forest in Greene. So we have a 142 acre model forest that gets a lot of people coming through hiking, and we're right at the base entering the Catskill Mountains. So
Right. Yeah, that's great. Oh, I should mention Meredith corrected me. So we've had a contract with CCE since 2011. I know we didn't have every year, like during the pandemic. We had to stop some of our subcontracts. So, but yeah, we really appreciate all the work that you guys do. All the outreach that you do on invasive species is really helpful and beneficial for all of us. Are there other questions that people have? For Becky? and you can unmute yourself if you'd like and jump in. Not hearing
I was
anything.
just, oh, I was just
Meredith,
curious.
go.
Yeah, thanks. I was just curious if the fair questions coming in this year, there was more public interest in spotted lanternfly, if that was a big one for you, or if it was just the same kinds of questions as in past years.
I would actually say that it probably was pretty much the same. So spotted lanternfly is one that we actually talk about and bring out a lot. So we tend to field a lot of questions. Like, for example, I don't think I've ever even mentioned this to John. I bring that up with me to the 4-H youth building at the state fair. I bring information about the spotted lanternfly because that seems to be one that's like resonating with people.
Mm-hmm.
So I wouldn't say it was necessarily any more than normal, Meredith, just because we tend to really talk about that one a lot.
Sure.
Other questions? Yeah, are there other species, I guess, that pops into my mind? Like, are there other species that you're seeing like come up in terms of public consciousness that they're asking you about?
It really varies by county and kind of what everyone's interest in. I know that we field a lot of questions about the things that are in our own forest. So I mentioned the stilt grass, and for some reason I am completely forgetting about what I want to say. Yellow iris was the other one. That might be wrong that we were pulling. It was yellow something out of the ponds over here. I couldn't join that day. So it varies greatly. What happens actually a lot with us is that people bring in plants for ID, and plants and bugs for ID.
Oh,
And
yeah.
so oftentimes that's where a lot of the questions come from, is that then we identify them as invasive species and start those conversations. I will be honest, and I'll have to follow up with Shane. I have not, since we started working, and that was new this year, we're really working with the master forest owners. I'll have to ask him what they have been seeing on their forest walks, because I haven't asked him, so to get an idea.
Okay. Oh, yeah, and I wanted to just check in with him about, we did the invasive species training, how many people they've been sharing that information with, if he could get those metrics back to us. That we were, the Catskill Center has that grant that they're working on, so that number would be really helpful. Any other questions for Becky? Otherwise, and I guess Meredith, if you had some photos you wanted to show or something that you wanted to share, you can go ahead.
Okay.
Other questions?
I did
Yep,
want to
go ahead.
catch myself. I double checked. It was yellow iris is the other one that we
Okay.
were really talking to people about here. Because we had a bunch of it. So it's always easier to talk to people about things that are in front of them.
Right. Definitely. Okay, thanks, Becky. And I think we'll go ahead. Meredith Taylor's gonna speak next. She's from New York City Department of Environmental Protection. And they've had a couple infestations of fish hook water flea that she wanted to update us on. So go ahead, Meredith. Thanks for joining us today.
Thanks, John. Is the map from IMAP showing up okay for you?
Yeah.
Okay. So I am not permitted to share a PowerPoint, but I will drop in some visuals for you. So this is the current distribution of reported fish hook water flea across New York in IMAP invasives. And you can see there's been a longer history of establishment in the Great Lakes and then Finger Lakes. The first detection outside of that region in 2018, I believe, was in Lake Champlain. And then the next detection was in 2023 in New Croton Reservoir, all the way down in Westchester County. So these are very broadly dispersed new detections. It's possible that there are other populations in between that we're not aware of, but it's also possible that folks that engage in some of these different fishing opportunities could be moving the fish of water fleet great distances unintentionally, and so they could be making these large leaps on fishing gear. So 2023, we detected it incidentally in a zebra mussel villager survey in New Croton Reservoir, just a few individuals. There was no huge population buildup that we could detect in our villager samples. It was just a few scattered here and there. And then we had this past summer in July, anglers reporting large numbers in Papacton Reservoir. At the end of the season, we had a detection of, again, just a few individuals that we were intentionally surveying for in Rondout Reservoir, which is connected via a tunnel to Papacton. So that's somewhere where we were expecting to find it. Just circling back, a little bit of background on fish of water flea, Cercopagus pangoi. It's a 1 to 3 millimeter predatory zooplankton, and it can have a 5 to 10 millimeter tail. The tail is part of the issue with this species because it does make it unpalatable to many fish species. So it's going to favorably shift the fish population toward species that can consume that tail. It's native to the Ponto-Caspian region, Eastern Europe, and it can reproduce both sexually and asexually. The end of the season, it can produce a resting egg, or like a cyst-like egg that can survive freezing and drying and ingestion by fish. And so that resting egg makes it very easy to transport large distances like we've seen in New York, and makes it really critical that we're extra vigilant on decontamination protocols to prevent further spread. So as I mentioned, we had the New Croton detection July of 2023, and then in July of 2024, reports began to come in from multiple anglers on Papacton Reservoir. They were seeing large clumps floating in the water, and their fishing lines were getting covered with the fish of water flea. And you can pull up this angler report. So it was found by a member of the public and reported into IMAP. Let me see, it's not shown. Quit right. I want the summary. Can I get the details? Nope. Just trying to get the photos to show up here.
I think you have to zoom in.
Oh, maybe that's the problem. Okay. Okay, so Papacton Reservoir has a large, deep section, and that is very much, there we go, details. And that is conducive to producing these large blooms of populations. We are being told that there's a clump of them, that the shallower waters can support small populations, but may not lead to these big clumps being produced in the same way. So there could be low-level infestations in many other water bodies, and it's not until it becomes invaded in these larger, deeper water bodies where the population can get to these high nuisance levels. And there's just a close-up on the hook there. So we did confirm the identification then that came in from angler reports, and then promptly began to survey the Rondat Reservoir, which is downstream. So the first detection there wasn't until September 3rd. We did some outreach via email blast to all of our permit holders in the impacted basins to let them know that it was present. We offered guidance on minimizing spread by engaging in decontamination of gear, and preferably encouraging them to devote specific fishing tackle to infested water bodies and not to move it to other water bodies in the region. We tried to do some additional outreach, but we had a lot of trouble. We couldn't get out of our own way in some ways to get more information out there. And we did go around to bait shops at the end of August, all of the bait shops in the region. We met with the owners or operators, let them know what was going on, left them some outreach material as another way to reach the anglers in the region. And just last week, we began our own in-reach effort, and we presented information on fishhook, water flea, and other aquatic invasives to all of our watershed maintainers in our regional offices throughout the watershed, so that they have the information they need to prevent the spread, and also that they can share information with the public when they interact with the public as they help folks get their boats out to the reservoir, or steam cleaning boats that are coming in, or interact in any number of ways with the public. So we're working on that. We've had a new sign order in for almost a year now. We've been trying to get new signage to go out, but we've had various contracting issues on the signage order. So we're still waiting for that to come through. But we're hoping to do some more outreach throughout the next few months to get the word out before the spring fishing season starts up again. On September 27th, we convened an expert panel to get input from researchers around the state who have worked on fishhook, water flea. We had questions that we wanted to know if anyone had background information on the impacts from fishhook, water flea on some of the top issues that we are concerned about for water quality, like dissolved organic carbon or natural organic matter, how fishhook, water flea might impact those parameters. We also wanted to know about how they have in other water bodies impacted the frequency of harmful algal blooms. We tried to get some input on how best to go about sampling for fishhook, water flea in the reservoirs that are known to be infested, and also those that are nearby, trying to detect it early. We asked if there was any other data we should be collecting to be able to assess the longer term ecological impacts. There are still quite a few unknowns with this species that hasn't been around all that long. And so some of the questions we didn't really get great answers to, but we did find out from what they've seen in Lake Champlain and other water bodies that population levels can vary from year to year. So where you might have a huge bloom in populations one year, the next year you could just find a few here and there. So that was good to know. The recommendations from outside experts really were to keep the focus on spread prevention, encouraging us to do whatever we could to keep gear and nets and boots separated from invaded reservoirs and uninvaded reservoirs. They recommended we go so far as to shut down any kind of access to the water bodies, which we don't really have the ability to do. We learned that we need to sample with a tow that goes rather quickly because they can swim out of the way. And we could deploy additional sampling tools such as sentinel lines, like fishing lines that could be deployed more widely throughout the reservoirs to be able to detect them. There was some skepticism about using environmental DNA because it can provide both a false sense of confidence in a negative detection, and it can also provide false positives if the samples are contaminated at all. We found out that there's lower population levels in shallower systems, and the populations are negatively impacted by turbidity. They are visual hunters, and so they can't find their prey in turbid waters, which could be of interest for us, with some of our systems much more turbid than others. The focus really was on getting some outreach out there, doing more of it, trying to really impress upon anglers the importance of taking these sort of extreme measures to stop the spread, which is a challenge, of course. And in soliciting feedback on how we might be able to do more sampling and better study these infestations, we received information about different groups to connect with. SUNY Plattsburgh offered to help with ID for zooplankton samples. They recommended reaching out to Cornell University's Shackleton Point Field Station, connecting with David Richardson at SUNY New Paltz, trying to get some support through the Carey Institute and the Catskill Science Collaborative. Daniel Stitch at SUNY Oneonta was interested in connecting with us for some work with his graduate students, and they recommended reaching out to the Citizens Statewide Lake Assessment Program, or CSLAP. So we have a bunch of different leads to follow up on now. We're trying to get a better handle on the distribution within our system, trying to come up with a sampling plan over this winter, and starting to dig into how we're going to assess our zooplankton communities to be able to detect changes over time as these infestations become established. So that was a lot of detail. If anyone has any questions, I'm happy to answer.
Thanks, Meredith. That's there's a lot, I guess there's a lot unknown, a lot to be done.
It is. It's kind of overwhelming how much we need to figure out over the next few months to get ready for the next season. The winters don't feel long enough sometimes, and sometimes they feel way too long. But yeah, we appreciate both John and Sarah participating in that expert panel and helping us as we sort through what to do in this situation.
Yeah, well, we're here to help, and we could definitely help with outreach, and something that because it is a new detection that we will incorporate into the outreach that we already do.
Great. Is there a management technique for fish hook water fleas? Unfortunately, no. The only real thing we can do is slow the spread. Any other questions? Do you have any idea what fish or organisms might eat it? Yes, there are a number of fish species that do consume it. I don't have the list at my fingertips, but I can share that. I believe largemouth bass may be able to, but not smallmouth. So there's definitely some variability between species that you might not expect, and it is primarily the young of the year fish that are going to be the most impacted.
Other questions?
Okay. Thanks.
Thanks, Meredith. We appreciate it, and good luck.
Thank you.
I think I'm next, and I can show the forest regeneration results. I can share my screen again. So as you are probably aware, can you see my slide? I just changed it to forest regeneration survey.
No, I don't see any
Nope.
slides, John.
Can you see it now?
Yep.
Yep.
Okay. So a couple weeks ago, we sent out a forestry regeneration survey that is based on the group concept mapping strategic planning that we did several years back. There was a statement that came up about forest regeneration. And so from that, we formed a forest regeneration working group and came up with a goal for this topic. And the goal is to rank the invasive species threats to forest regeneration and forest ecosystem services and their mitigation strategies, identify research needs to guide future CRISP RFPs, grant applications and other research initiatives, and CRISP work planning. So that's why we did the survey. And in the survey, slides again. In the survey, we asked people what they thought were some invasive species that had negative impacts on forest regeneration. And this is the ranking of those species. Stillgrass came out first. Barberry was pretty close to that. And then bush honeysuckles, bittersweet, multiflora rose, and burning bush. So it's basically the herbaceous stillgrass that people ranked most highly, and then several shrubs, then the bittersweet vine, and then ending with multiflora rose and burning bush. Then we also looked at, I can get my slide to change. What are the forest pests and diseases that are having the most impact? Emerald ash borer came out as the #1, followed by hemlock woolly adelgid, jumping worms, beech bark disease, spongy moth, Dutch elm disease, and chestnut blight. That was, I think it was a little bit surprising to me, but that's the results that we came up with. And I should say too, I'm just gonna kind of mention the results today. We'll be looking into them more closely over time. We just closed the survey the other day, so there's lots of results and fill in the blanks that we need to analyze and look at and kind of digest over time. So some of the emerging impacts of invasive species or emerging forest pests that we saw or that we asked people about were beech leaf disease was ranked #1, spotted lanternfly, then southern pine beetle, and then elm zigzag sawfly. So beech leaf disease will be interesting as it continues to spread in our region. Pretty much, well, it started in 2022 and it's pretty much spread over the whole entire blue line, most of the CRISP region, except I don't think, at least when I last looked, there wasn't a observation in Schoharie County. So, but I expect at some point there will be. Then scanning the horizon, what are some of the species that people thought would have the most impact over time? Asian longhorn beetle has always been something since CRISP started, has been a really important species that we focused on. Oak wilt came in second, then sudden oak death, and then feral swine. So those are the species that are on the horizon that aren't here that could have a big impact on forest regeneration and these forest processes that we were asking people about. Then the next question, and this will be the last one that I'll talk about today, barriers to mitigation. What do people think were some of the barriers that are preventing us from doing mitigation on the ground for forest regeneration? And tools and equipment came out first, then access to properties for management, then access to knowledge about these invasive species or tools to treat them. Volunteers came out next. Availability of pesticide applicators was after that. Access to native plants to plant. Then competing priorities that we all have when trying to address invasive species. Then we had to fill in the blank for others, which we'll be looking at over the next few weeks, and we'll try to come up with some summary of the findings that include the other. And then the final thing was consensus among colleagues. That was the lowest ranked thing. So those are the results of the forest regeneration survey. Do you have, if anybody has any comments about, and thank you all. There's a number of people on here that filled out the survey that we sent out. We had kind of a limited timeline where we wanted to capture that information. And I appreciate those that filled it out, getting feedback to us about it. And then, of course, the reason that we're working on it is that we've identified this as an important priority to work on. We have the working group that will kind of be digesting the results and coming up with other ways to look at what is going on the ground in terms of our forest processes and how to best try to address the impacts from invasive species and other impediments to forest regeneration that are happening on the ground. Does anybody have any comments or something they filled out that they wanted to point out, or any questions about that? And you could either enter it in the chat or unmute yourself, and I'd be happy to chat with people about it. I'm not hearing anything. Well, like I said, we'll be working on summarizing that information. We'll put a little report together. We can share it on the listserv. Probably have maybe we could put it up on the website, and so it will be available to everyone. But definitely could make it available so that people could see it and help us to try to answer some of the questions that have come up. And address these concerns over time. So I think next one, can't see my agenda anymore, but I think the next thing that we have is partner updates and announcements. And I know that I see Jonathan on here. I know that Radka had said that you wanted to share something, Jonathan.
Yes. Yes.
Oh, there you are.
We do. Sorry, we were muted and obscured. That's the edge of Radka. There's the rest. Yeah. So we would like to, if possible, share some slides and some updates on Mama. If
Great.
that's okay. Should we go
you should
now?
have
Okay.
permission. Yeah, you should have permission to share.
Okay, here. Yeah. All right, we're about to share. And
Is this it, right?
Yes, that's it. Chris Mama update. Okay, great. So, yes, or it's not, sorry, it's not cooperating here. Here we go. But we have to go page up. Okay, great. So we're just gonna share a few slides here. And thank you for giving us the opportunity to do this. So Mama, as you'll see, the Monitoring and Managing Ash program, really one of the first places it started was in CRISP, thanks to your interest, John, and other folks involved in the PRISM. And it's grown considerably since then. That was back in, I think, 2017, 2018. And I'm going to just kind of, we're going to update you on. And now, I don't think it's being shared. Because, it's really weird. Okay, so I saw that there are a few of you there who I don't know if you're familiar with CRISP or not. We have some diehards who are familiar with Mama or not, who have been involved since the beginning. And so we're just gonna maybe whip through some background really fast so this makes sense to you. So it's just a program. Mama is a program of ERI that enables detection of lingering ash through data collection and encouraging management practices that facilitate lingering ash detection. And what are lingering ash and how are they used for resistance breeding? So lingering ash, very specific definition, are mature trees. They're at least 4 inches in DBH. They're naturally occurring. They're chemically untreated trees that remarkably stay healthy for at least 2 years after at least 95% of nearby ash have been killed by EAB. They've been found for all 3 widespread northeastern ash species, white, green, and black. And here you have a photo of a lingering white ash that we found either in the Catskills or the Hudson Valley. And the U.S. Forest Service EAB resistance breeding project has shown that scion, in other words, twigs collected from lingering ash, can be used in selective breeding by grafting them onto rootstock and then doing successive rounds of breeding to yield highly EAB resistant trees. And Mama has been incorporated into broader regional initiatives. The Tree Species in Peril initiative, which covers basically the whole northeastern U.S., is led by the Nature Conservancy in collaboration with the Forest Service, the Sustaining Ash Partners Network, SAPNI, the Forest Ecosystem Monitoring Collaborative, FEMC. So I'm sharing this not so much to brag about where we are right now, but also to give your PRISM credit for really giving us our start in this. And so there are 4 Mama data projects, the Mama Ash EAB surveys, where you just report EAB evidence. This has become really important now that the agency, DEC, is really, for the most part, not tracking the progression of EAB anymore across the state. And it's certain there are areas that are long infested and areas yet to be infested. Mama Monitoring Plots Network, monitoring ash mortality using tagged trees. A fairly new project we have called Mama Rapid Ash Mortality Assessments, also known as Mama Ramas. And these are quick evaluations of sites that show some mortality where you then quantify that mortality. And finally, the lingering ash search, reporting of lingering ash in high mortality areas. So here we have one of our action maps, and this happens to be of New York and the adjacent states. And you can see this, let's see if I can get my cursor to work. I can't, my pointer. But if you see down in the, kind of southeastern portion of the state, you see that big red blob, which is portions of the Hudson Valley and adjacent Catskills. And that's an area that's been really, really long infested, and where there's high enough mortality for a long enough time that we can find those lingering ash. And then there are other areas, even in the Catskills, for which EAB has not yet even been detected. Just to show how far the program has progressed, this shows monitoring plots, lingering ash found, EAB ash surveys, RAMAs, and so forth throughout the Northeast. But the Catskills and Hudson Valley are still the core of this project. So we've already found about 180 lingering ash in New York and adjacent Connecticut. I should mention that only one of those so far is in Connecticut, because we just recently expanded there. but lingering white ash, lingering black ash, and lingering green ash. And the black ash are really kind of the big surprise and the great news to have found those. And here are examples of a couple in the Catskills. This is one near a mama monitoring plot right at the Catskills Visitor Center, and you can see that tree is really thriving. And virtually all the other ash at the site are either dead or very close to death. And then we have at lingering white ash near a mama monitoring plot at CCE Sullivan County, and there the mortality rate is about 93%. And with the remaining trees being in really bad shape, except for this one, which is also thriving. Here we have a picture of collecting scion, which has to be done in the winter when the buds are dormant. And so there's Radka using what's known as a big shot slingshot to collect scion from a high branch in this white ash tree in, I guess, in the Rosendale area. And those are then shipped to the Coldwater Pond Nursery, where they graft them onto green ash rootstock and sent them to Cornell for resistance breeding. So the grafts of the lingering ash that were collected through the MAMA project showed a success rate of 93% in terms of taking onto the rootstock, which is an exceptionally high percent, and it's just everybody who's been involved in this has been really diligent in doing the best job. And so now for basically each one of those lingering ash from which we collected scion, so out of those 180, we've collected scion from, I think, 26. Each of those, we now have 5 replicates, 5 trees that have identical genotypes to those lingering ash, and those will be used for resistance breeding. And then just a couple of other things that we've been working on. Our big thrust now is in not only in building, increased data collection, but really in incorporating lingering ash detection into management. So managers can enable lingering ash detection through data collection by setting up plots, etc., by leaving enough standing healthy ash, i.e. avoiding excessive proactive cutting, that some might turn out to be lingering ash. So depending on the species and the location, lingering ash can be much less than 1% of the population, or sometimes they can be, even more than 1%, maybe 1 or 2 or 3%, something in that area. And if you don't leave enough healthy ash at the beginning to, for some of those to ultimately materialize as lingering ash, as the vast majority of the trees die, you'll never find any. Think of it, if it's like 1 out of 100 trees, you're not going to have reasonably a chance of finding one unless you leave at least 100 that where maybe 99 of those will die and reveal that one healthy ash. And just more generally, harmonizing lingering ash detection with other management tools, which includes, for instance, chemical treatment, parasitoid release, seed collection, other silvicultural practices, etc. These can all be woven together and complement each other. And next, some other just interesting findings, which is, as I mentioned, so some places in New York, including in the Catskills, still don't show EAB infestation. So we have lots of plots in the Catskills, largely thanks to a CRISP over the years. And some of these still have not shown any sign of EAB infestation, which is kind of cool and encouraging. These can be places that are, 10 or 20 miles down the road from areas that are heavily infested. And it means that we shouldn't be in too deep of a panic about it. We should be appropriately calibrate our concern about EAB, but there's the misconception that everything is infested and that everything's dying or everything's dead, and that's not the case. We've also found lots of variation in mortality trajectories, with white ash in some places showing very slow stand level mortality progression. So some places in the Northeast, we're finding that it can take 20 years to reach really high EAB-induced mortality in ash plots. And I think the common idea is that basically uniformly, within 5 or 6 or 7 years, all the trees will be dead. That isn't necessarily the case. And so that can have management implications. It can change kind of the management timeline. On the other hand, there are some places where the ash die especially quickly. So probably genetics are involved, as well as environmental stressors or the lack thereof. And then since we have this opportunity to reach out to all of you, what we're really looking for, in terms of more involvement, is for people in the Catskills to do the Mama-Ramas. These are the rapid mortality assessments. You can get a few of those done in a day. You can see the stand of ash along the side of a road and go out and assess their mortality. That's a lot faster than doing a mortality monitoring plot. It's a one-time assessment, and it doesn't involve tagging trees, and it's done a little more coarsely, but it gets us really important information that fills in the gaps on our action maps. We're looking for people to do systematic lingering ash searches in those areas that are shown by the action map to really have high mortality already, and to validate reported lingering ash. So this wouldn't be, most of these would not be doable until the subsequent field season, summer of 2025. But we'll have folks reporting lingering ash to us, and sometimes they get the species wrong, or the tree isn't quite that healthy, or the mortality level of the surrounding trees isn't that high. So we need people to go out and validate them. And finally, this is a really cool thing that folks can get to do, is collecting, but it has to be through us and through our supervision and so forth. We don't want people going out and collecting cyan on their own, but we can train people to do cyan collection, because it's likely that as the long EAB invaded area in the Catskills expands over the years, that we will be needing to collect cyan from lingering ash all across your vast region. And we can't go out and collect all the cyan ourselves. So we have a few lingering ash to collect from this winter. If anyone's interested, they can potentially come along with us. We can show them how to use a big slingshot, how to prepare cyan for shipping and send them to be grafted and so forth. So if anybody is interested or wants to help us recruit folks for these tasks, we would greatly appreciate it. And want to thank Chris for all their support over the years. And thank you for inviting us to share this information.
Great. Thanks, Jonathan. That's really great. It's really amazing. You said 180 lingering ash in New York. That's great. That's incredible. And your grafting success is really encouraging. Jack Tessier has a question there. Do you want to address it, Jonathan? Do you see the?
Yeah, sure. Is it in the chat?
It's in the chat.
I'm sorry. Somehow I'm stopping the share so I can see the chat. Thank you. I was contacted by a local electric cooperative about collecting ash seed to store until after EAB dies down. I'm wondering if it makes sense to wait to collect seed until we can identify lingering ash and collect seeds from them. Thoughts? So two thoughts. One, there are initiatives to collect ash seed just to preserve as much genetic diversity from ash as possible. So those initiatives are underway, and that can make sense to do. And if it's a big mass year for ash, to go out and collect a lot of seeds, and those can be used in various ways. The other thing you can do is, once lingering ash are identified, in addition to cyan being collected from them, seed can be collected from them and identified as lingering ash and used for selective breeding. The only thing is, though, that there's a big advantage in doing breeding from cyan opposed to seed, because the seed are getting 50% of their genetic material from the mother, which is lingering, and 50% from the father, and you don't really know about dad. Dad may be a highly susceptible tree. So it still helps. It's going to have probably a higher level of resistance than just a randomly collected seed from a randomly found tree. So I would suggest to go out, if you have the resources to do it, if you want to do seed collection now, go ahead and do it. That can be used in some really useful ways in terms of genetic diversity conservation. But then also to target lingering ash trees. I mean, you're not going to have like a zillion lingering ash trees out there. When we're looking at these 180 trees, they're scattered across a fairly broad area of the Hudson Valley and some part of the adjacent Catskills. It's still a small area of New York, so we anticipate to find many, many more over the coming years. But I think it's fine to go ahead for now and do general collection. And once you find out about lingering ash in your area, you can also target those. But keep in mind that, it may be that less than 50% of the ash trees, in your area to begin with can be female, right? And so to find a female tree that's healthy, that's bearing seed, that's lingering and so forth, I wouldn't save all my energy for then. If you want to do seed collection, it's fine to do it now.
And then Mark?
I see. So I see, why is green ash the choice for the rootstock? And a couple of reasons. One, it's what has been commercially available, and that's because green ash is being planted really widely in cities and towns and parks and people's front yards and so forth. So our partners have all been using commercially raised green ash rootstock. Which is actually an issue because that's getting harder and harder to obtain. There's basically one nursery now in the country that's still producing that. And how much longer they'll do that, I don't know, because there's not a lot of money to be made in raising green ash rootstock. And the other part is that green ash is kind of like the medium species. If you think of it, there's green ash, white ash, and black ash. And white ash likes the drier circumstances. Green ash kind of the middle, like the floodplains, and black ash really likes to grow in swamps. So green ash, by having that rootstock, you can use that to graft all three species on and then plant them in areas that can be suitable for those different species when you're then growing those trees out. But we're actually looking into, because what we're finding, so white ash are much more abundant than black ash or green ash in our part of the state. And so what we're thinking of doing going forward is actually here, for what we're doing here, is using white ash rootstock and potentially partnering with some folks who are interested in actually doing seed collection and growing rootstock from local white ash that we can then use for rootstock. Any other questions? Great. Thanks again.
Yeah, thanks, Jonathan and Radka. Really appreciate it. That's great work.
Thank you.
Does anyone else have any other updates? Comments?
Hey, John, it's Mark Wolf.
Yep, go Mark.
I could just give quick updates on what we're doing at Mountaintop Arboretum. A lot of it's just continuing. We do have, Jonathan, I don't know if you know about it, we have the Emerald Ash Borer study we're doing with Mark Whitmore at Cornell that we're wrapping up now. And we just got a private donation to continue the work ourselves for another 10 years, where we're treating the ash, and we'll continue studying them, but it won't be as, scientifically set up as Mark Whitmore was doing, because we don't have that background. We're also working with DEP. Meredith helps, and John helped set that up. We're working with DEP on permits to treat for hemlock woolly adelgid in our 156-acre easement. Lesser celandine, we're digging in the spring in areas where we feel we can control it, and we experimented with planting plugs to sort of hopefully outcompete it. We planted some aggressive native plugs. Marsh thistle, we're using a protocol from Donna Vogler that we're tweaking a bit to limit that. And honeysuckle, we were looking for maybe less toxic ways than cutting and painting, which is pretty targeted, but we're still always looking for ways. And I reached out to you, and you said you were looking at buckthorn bags, and Dan on Beauty Bush, I think. So we're going to try buckthorn bags on a section of the honeysuckle and let you know how that goes. So I think that's it. What species are the plugs? What was the next part of that question? What's this? We're using Senecio aurea, a cultivar of Carex grayi from New Moon that they're calling it like New Moon or something because it's a bulkier, more aggressive spreader than the species. And oh, there was another Carex that I'm blanking on, but you could reach out to me, Meredith, or anybody else. They all came from New Moon. Yeah, I'm blanking on the other species, but I'll send it to Meredith, and anybody else wants to know, just email me, Mark. I'm at Mark at marc@mtarboretum.org.
And Jonathan put his email address in there for you, Mark,
Great,
in the
thanks.
chat. Does anyone else have any quick updates? I have another meeting at 3 o'clock, so I'll need to jump in a few minutes, but we do have a minute or two. Otherwise, I could wrap things up. Not seeing anybody. So yeah, I just want to thank again everybody who participated in the meeting, especially Jeff, Becky, Meredith, Jonathan. Thanks for everyone that contributed. I really appreciate it. Our next meeting is going to be on March 27th on Zoom. The registration link is at the bottom of the agenda that I sent out. We'll be posting it on the CRISP website under events. And I hope that everybody has a really great and happy holiday season, and really appreciate your time and meeting with us today, and look forward to seeing you either later this year or in the new year. Thanks a lot.